I am Lishma, Assistant Professor, Department of Microbiology at Al Jamia Arts and Science College. Today, we are going to discuss about the simple and widely used staining procedure, that is gram staining. It is one of the most common and universal staining procedure for the identification of bacteria. This staining procedure differentiates bacteria into two large groups such as gram-positive bacteria and gram-negative bacteria. Valuable diagnostic tool in both clinical and research setting. Not all bacteria can be definitely classified by this technique. This gram staining procedure is invented by Danish scientist Hans Christian Gram in 1884, and this technique is modified by Huckers and Kohns in 1921. The agents used in this staining technique includes the primary stain, crystal violet, and the modern grams iodine, and third one, decolorizer. It is 95% ethyl alcohol or acetone is used as the decolorizer, and last one, saffron. It is the counter stain used in this gram staining procedure. Then the procedure of the staining procedure. First, take a grease-free clean glass slide and prepare a thin smear of the culture. Then gently fluid the smear with the crystal violet, that is the primary stain, and let it stand for 1 minute. After 1 minute, gently wash with distilled water or tap water using a wash bottle. Then after that, gently fluid the smear with gram iodine, that is the modern used in this staining procedure, and it also let it stand for 1 minute. Then after using the grams iodine, the modern, gently wash with distilled water or tap water. And decolorize using 95% ethyl alcohol or acetone, and let it stand for just 5 or 10 seconds. Then immediately rinse with the distilled water, and after that we can counter stain with the saffron. It is a counter stain used in this staining procedure. Let it stand for 30 to 45 seconds. After that, we can also wash with the distilled water. Then air dry the smear, and we can observe using a light microscope under oil immersion objective. This is the procedure of the gram staining technique. This is a pictorial representation of gram staining procedure. It includes application of crystal violet. The cells will appear as purple color. Then application of application of grams iodine, the modern, and third one, application of the decolorizer, alcohol or acetone, and fourth step, application of the counter stain, saffron. When we are going through the principle or mechanism behind the gram staining procedure, first we are using purple colored crystal violet as the primary stain. All cells become purple in color. Then we are applying the modern amber colored grams iodine. It attaches to the primary stain crystal violet and forms a large complex that is called a CVI complex. You know that in gram-positive bacterial cell wall, it is very thick and it's made up of highly cross-linked peptidoglycan layer. And in case of gram-negative bacteria, the peptidoglycan layer is very thin. So when we are applying the decolorizer ethanol, it will remove the stain only from the gram-negative bacteria, because the peptidoglycan layer is very thin, and it removes the CVI complex from the gram-negative bacteria. And after that, we are applying the counter stain, saffron. It is a red or pink colored stain. It stains only the gram-negative cells. As a result, we get the gram-positive bacteria in purple color and gram-negative bacteria as pink or red color. Examples of gram-positive bacteria include Actinomyces, Bacillus, Clostridium, Corynebacterium, Enterococcus, Gardnerella, Lactobacillus, Listeria, Mycoplasma, Nocardia, Staphylococcus, Streptococcus, etc. The examples for gram-negative bacteria includes the common Escherichia coli, Salmonella, Shigella, and other Enterobacteriaceae, Pseudomonas, Moraxella, etc. These are all about Gram staining procedure. Okay, thank you.